Arunava is a master's student studying in France, and he's also a visiting scholar in Blue Marble Space Institute of Science. Today he's going to tell us about stable liquid-liquid phase-separated systems formed by intrinsically disordered proteins. We can't wait to hear what have you prepared for us. So Arunava, I'm giving you the floor.
Thank you, Rosa, for the great introduction. So I'm going to share my screen.
You should see an option to share screen in the like probably beyond your oh, okay, great, you can see.
Is it visible?
Yes, it is. Yeah, great.
Okay. Thanks. So hi everyone, I'm Arunava, and again I'm a visiting scholar at Blue Marble Space Institute of Science. And it's a great honor to present my work in the Life and Space conference today. So today I'm going to talk about intrinsically disordered proteins in phase separation from origins of life to modern cells. So this is a review that has been done by me and my colleagues under the supervision of Dr. Tony. So you guys might be familiar with the Oparin-Haldane hypothesis, which states that conservates or droplets encapsulating life-relevant molecules arose from a warm little pond or a primordial soup of sorts. And these droplets basically evolved into all life as we know it on Earth. So these droplets basically separated themselves from the rest of the environment. And this phenomenon is nothing but phase separation. Now, phase separation involves one or more polymers. And as you can see in this figure out here, there are two polymers indicated in green and purple. And so if these polymers segregate amongst each other, they form segregative liquid-liquid phase separated systems or segregative LLPS systems. But these don't form conservates. But if these polymers associate with each other to form complex conservates, then we basically get this kind of a conservate. Another interesting fact out here is if only a single polymer is involved, then that polymer is going to interact with itself or with each other of the same type to form simple conservates, as we can see here. Now, something to note out here is these polymers and biomolecules are encapsulating a lot of enzymatic reactions without the requirement of a membrane. Which brings me to the question, are all cell organelles bound by membranes? Well, the short answer is no. Contrary to the popular belief, there are a lot of cell organelles which do not have a membrane. So here you can see a eukaryotic cell, and all the organelles that are bound by that are not bound by membranes are indicated in dashed lines out here. So you can see so many of them out here. These organelles perform different types of functions like RNA splicing, ribosome synthesis, stress regulation, signal transduction, etc. These organelles are composed of a particular type of protein known as the intrinsically disordered protein. So these proteins are not our usual structural proteins that we mostly know of. As you can see in these GIF images out here, these proteins are kind of floppy, right? So they don't have a particular conformation at a given interval of time. The conformation changes at regular time intervals. And that's why they do not have a fixed order structure. Also, they are composed of charged residues, and they are of low complexity, which means that they are composed of limited amino acids out of the 20 that we know of. Similarly, when these proteins associate with each other or with RNA or DNA, they can actually attain a stable conformation, a stable 3D structure. And as you can see in this image, this protein, which is intrinsically disordered, is bound to DNA. And after this binding, they can attain a stable 3D structure. And finally, these disordered proteins contribute to the formation and function of membraneless organelles. So let's look at the work that has been done before. So Sidney Fox was the first researcher to synthesize proteinoids under conditions similar to primitive Earth. Now these proteinoids are nothing but proteins which are similar to disordered proteins. And these proteins can form microspheres when they are dissolved in water. Now microspheres are just small water droplets. Now, proteinoids also contain mostly charged residues and are mostly of low complexity. And this is exactly why it has been hypothesized that proteinoids were precursors of disordered proteins, and primordial disordered proteins from these proteinoids. And the work that has not been done is a proper comparison between modern disordered proteins that forms membraneless organelles, just like how we saw, with primitive peptides that probably form primitive conservates on the basis of protein amino acid composition and length. So the hypothesis out here is are disordered proteins that form membraneless organelles in cells somehow related with primitive IDPs that formed primitive conservates, and we are going to see how they relate with each other. So first, let's look at the comparison of amino acid composition between peptides that formed primitive conservates versus those which form modern membraneless organelles. So before delving into the topic, I should mention the fact that all amino acids did not evolve at the same time. So aromatic amino acids like phenylalanine, tyrosine, tryptophan are structurally very complex, and they have a higher molecular weight. And these evolved later during the time span. And these aromatic amino acids actually contribute to structure in structured proteins. Now, the late evolution of these aromatic amino acids indicate the fact that primitive proteins that evolved before the evolution of these aromatic amino acids were probably disordered. This is also supported by the fact that modern IDPs have limited aromatic amino acids, and they are mostly composed of charged amino acids. So now let's look at the role of charged amino acids in primitive and modern phase separation. There are certain types of charged domains in these disordered proteins known as the arginine, glycine, glycine domain. And so these form different conservates without the requirement of the entire protein. So you just have these RGG domains and you perform phase separation studies, and you will see different phase separated conservates without the requirement of the rest of the protein. So this is pretty interesting, and it has also been hypothesized that these domains have evolved early and would have contributed to primitive RNA metabolism and folding. These studies indicate that short disordered RGG domains or RGG containing peptides would have formed primitive conservates. Let's look more on the role of charged amino acids. Along with these RGG domains, there are certain homopeptides like polyarginine or polylysine that are associated with these. And these are also found to be unstructured. So And also, these homopeptides, interestingly, are also used in primitive conservate studies in vitro. So this is pretty interesting because, as we can see, charged amino acids have a huge role in the formation of modern membraneless organelles, as well as primitive peptides that could have formed primitive conservates in vitro. And finally, let's look at the role of non-canonical amino acids in these conservate formation in relation with modern proteins. So non-canonical amino acids are the ones which are not present in modern proteins, but are thought to be present in primitive proteins. So there was a study which involved a protein motif known as the Triple H motif, which can bind to DNA. And this particular motif was ancestrally reconstructed by replacing the lysine residues with ornithine. So ornithine is a precursor of lysine, and it is also non-canonical amino acid. So the resultant protein that they obtained is kind of similar to a primitive protein, and this was found to be disordered as compared to its parent motif. So when this disordered primitive protein was associated with poly-U RNA, it was shown that it can form conservates or droplets. And this is extremely interesting because this means that ancestral disordered peptides containing non-canonical amino acids would have formed primitive conservates. So now let's see how lengths of different disordered proteins or peptides that formed primitive conservates is somehow related to modern membraneless organelles. So talking about structural proteins, a particular structural protein attains its structure by folding itself into a conformation which has a hydrophobic interior and a hydrophilic exterior. Now, this particular conformation is possible only when a particular protein is quite large. Now, this possibility of producing a large protein in prebiotic earth conditions is not that much, because if we consider the conditions of primitive earth, they were not very suitable for the synthesis of large proteins. Now, this is pretty significant because this means that formation of short peptides on primitive earth would have given rise to proteins without folding capabilities. And indeed, some minimalistic studies have shown that small peptides can actually form conservates. And this can be correlated again with a previous study where we saw that short RGG domains alone, without the requirement of the entire protein, can phase separate. Finally, to conclude, I would say that this review was an attempt to bridge the knowledge gap between primitive and modern phase separated systems with respect to disordered peptides. And the knowledge gaps that we have tried to address is the fact that primitive conservate formation has not been much explored in the context of primitive disordered peptides. And also primitive conservates have not been much correlated with membraneless organelles, just like how I discussed. And finally, ancestral reconstruction of IDPs that form membraneless organelles to produce prebiotic disordered proteins, and testing their ability to form conservates might answer some of the questions associated with primitive conservates and membraneless organelles in modern cells. So our group is also trying to find new similarities or dissimilarities between different membraneless organelles and primitive conservates. And so I would like to thank Dr. Tony, who is my mentor for this project, for his immense guidance, and all my fellow visiting scholars who have helped me a lot throughout this study. And so thank you. I will be ready for any questions. Thank you. I'm sorry, I think I can't hear you. Yeah.
Okay, maybe that would be better.
Yeah.
Okay, I'm not sure why it isn't working when I try to unmute for the first time, but okay, I'm glad you can hear me now. So thank you so much for your very interesting talk and for perfect timing, because it took exactly 15 minutes. So we are waiting for questions from the chat, and in this time I will have one question. You mentioned proteinoids, which are precursors of IDPs, and they do form microspheres. But they, as I understand, they do not form liquid phase separated systems. So what is the actual difference between proteinoids and IDPs? I mean, what happens when proteinoids is becoming like a okay,
actually,
you understand the question. Mm-hmm.
yeah. Yeah, so actually, the first study that was done is associated with proteinoids. So at that time, people were not really aware about disordered proteins, and uh, those proteinoids can actually form microspheres, but actually, it's a form of phase separation, but it has not been quite associated with the modern membraneless organelles that we see in cells these days. So the studies with proteinoids like is kind of like these days the study of proteinoids is kind of done with respect to disordered proteins or polymers of that kind of conformation, but protein, they're not called as proteinoids, but disordered proteins and other kinds of polymers are used to understand phase separation, and they are used to kind of try to understand why phase separation happens in our cells as well as in normal other conditions.
Okay, thank you very much. We have also a question from the chat from Matthew Mersky. If you're thinking of the exacerbates as a phase change, have you looked into what the actual phase diagram is like for them?
I actually have not looked much with respect to phase diagram, but I can say this, that the phase that is present inside the conservates, we can't, it's actually liquid, and that is why we call it liquid phase separation. The environment outside it is also in a liquid state, as well as the environment inside the conservates is also liquid. It's just that the conservate contains a higher concentration of biomolecules, and that is why it basically separates itself from the rest of the environment. We can think of it as a difference in density. It says that this is different because biomolecules basically form like kind of a aggregate which separate themselves from the rest of the environment. So yeah, that's it.
Okay, so thank you so much for the answer. And I think we can jump to the next speaker. So thank you once again, Arunava.